In this lesson, we're going to see how the body fights back against viruses. And you're going to recognize some of the information. A little bit's going to be review. We're going to see some sensors we've seen before, types of cells we've seen before, but there will be some new information as well. And I'd like to make a huge disclaimer right here at the beginning that this is not a complete lesson on immunology. There's things that I'm omitting. We're not discussing the whole immune system. This is just a few highlights, and particularly the things that are related to viruses. There are basically 3 levels of defense. The first is physical barriers. We try to not let them get in to begin with. But then if some of the viruses get in, we have another 2 layers, and the first one is your innate or non-specific immune system, and that'll be number 2 and 3 and 4 and 5. And then after that, we have the adaptive or very specific system that goes after particular viruses, and that'll be section 6 and 7. But before we start drawing our first physical barrier, it's going to be skin, let me show you a picture. This is a very simple diagram of how skin cells are arranged. And this layer on the bottom, the basal layer, these are the living cells. These are the cells that are dividing all the time. They constantly make copies, copies, copies. And the copies get pushed up because they're always piling up from the bottom, and the pile gets pushed up higher and higher. And as the cells rise and they get further away from the living cells, the cells start to die. But that's okay. In this case, it's a good thing. You see how the nucleus is getting smaller here, and all the organelles break down. They get thinner and smaller. And by the time they get to the top, they're completely dead. No nucleus, no ER, no Golgi bodies, no mitochondria. They're just kind of basically dead shells made out of protein. But because this outermost layer is dead, that means that viruses can't attach to it. Viruses have to have those cell receptors. These cells down here have receptors, but these guys are dead. Their receptors are gone. And so viruses can't attach to this layer. So let's draw a little chunk of skin, very much like that diagram we just saw. I'm going to make my label going top to bottom. We have a limited amount of space here. So I'm going to write the word skin like this. And I'm going to make that bottom layer. It's nice because these cells are actually pretty square. Makes them very easy to draw. Just draw a layer of square cells like that. And then make sure they all have a nucleus. And then if you want to add some of the little squiggle, just so they look like they're full. They're healthy cells full of organelles. And then we'll draw another layer. Still pretty healthy, pretty living. Still put some nucleuses in them. And then another layer. And the layers get a little bit thinner as they go up. Maybe still a little bit of a nucleus there, although it's just starting to disintegrate a little bit. Even thinner. So this top layer is the stuff that's sloughing off all the time. Every time something touches it, a few of these little flakes come off. I'm just going to write here, dead cells on top. So with this, we've taken care of all of the external parts of the body. But what about the parts like our mouth, our nose, our throat, our intestines, which definitely come into contact with the outside world? They're the outer layer, but kind of on the inside. But they're still epithelial cells. You take a big breath of air, and the air goes into your lungs, and those cells come into contact with that air. So those cells, almost all of them, produce mucus. You also have saliva in your mouth, but we're going to talk about the mucoid layer. Let's just write mucus. mucus. So even in your intestines, the cells on the surface are producing a mucus layer. You know, we know there's mucus in our nose and our sinuses, but you have it inside as well. So here's a diagram showing some of the cells in the top part of the mucosal layer. These orange ones here, these are like the regular cells, and they have this little wavy border. The cells, especially in the intestine, they have these little, like fingers, are called villi. So those are actually part of the cell. And wedged in among these intestinal cells, we have specialized cells. This one right here is producing lysozymes. That's a word we've seen before. Those are antimicrobial enzymes that can break down things in bacteria. Those are antibacterial. And we have these type of cells called goblet cells, which produce the mucus. So that's the mucus producing one right there. And then the mucus would just kind of go out and cover the top layer. They don't show the mucus layer here. And here's a type of cell we'll see in a minute. It's called a dendritic cell, and it has very long arms, and it can actually reach up between the cells, reach its arm out and grab a bacteria or a virus sitting on the surface. Pretty cool. Here's another kind of cell lurking on below, macrophage. We'll see that one also. And here's a cell sitting down here, a B cell. We'll see those at the very bottom of our drawing. It's making these little antibodies. These are IgA antibodies that are going up between the cells and out, and they're sticking to pathogens out here. And here's another immune cell sitting in here, producing things that are going to go out and attack the pathogens. So we definitely don't have a dead surface here. This is much different from skin. So because it's not dead, we need a lot of protection going on. Now we're going to make a very much simpler version of this. Let's draw a couple of tall cells like this. Put a nucleus in them. And then we'll draw a kind of different looking one that's like a little more round or something. Pretend that's our goblet cell. The regular cells tend to have this little things on the end like that. Okay, and there's another one. And then we'll do another specialized cell. And the point of this drawing isn't to find out what all these cells do. We're just going to say that they are antimicrobial germ-fighting cells. And this cell right here, the goblet cell, is going to be producing mucus. I'm going to use yellow. And it's going to go out and have a nice protective covering. And of course, you've got lots of these goblet cells. I'll make this one, I'll make it all yellow. You can tell where it's coming from, that cell. Of course, in your nose and throat, you've got lots and lots of these protecting inside your nose. And hopefully what's going to happen is that if a virus comes along, it'll just get stuck. Now I'm going to choose a color that I'm going to stay with for the whole drawing, and everything related to a virus, little bits of protein or whatever, something, I'll stay with that color. You can choose whatever color you want. I don't know, I'm going to choose purple right now. So I don't have to label these. I know these are viruses. So if they land, hopefully they'll get stuck in the mucus, and then the mucus is constantly flowing and washing things out. You've got all those little IgA antibodies. So hopefully that's going to rinse out a lot of your viruses that way, and they'll never get close to the cell. They'll just get washed away by the mucus. So these would be cells in the, see, nose, mouth, throat, intestines. Of course, the eventual fate of this mucus, if it's in the nose, throat, lungs, it's going to get coughed up or sniffed or swallowed. It'll end up in the stomach where there's very strong acids that will hopefully digest all the little pathogens. And if it's in the intestines, then it'll just go out with the waste material. So the mucus will eventually end up getting flushed out, and hopefully these viruses will just get flushed out along with the mucus. So if the virus makes it past those physical barriers and it gets in, it's going to meet these immune cells. This one and this one are the ones we're going to draw. And they are what I've called the roaming eaters, phagocytes. Phago means to eat, and their job is to go around and eat all the little pieces of stuff they find lying around. Maybe it's little bits of dead cell, little cellular garbage, or foreign objects that shouldn't be there. Anything that doesn't belong, these guys will eat it up and try to digest it. Now they have really blobby shapes, so you don't have to worry about making your shape look just like mine, because their shape changes. They're kind of like an amoeba, if you've ever studied protozoa. They kind of ooze around and their shape can change a bit. So we're going to draw two cells, one here and one here, just so you can kind of leave space for the other one here. And this one's just going to be kind of an oozy shape. But as you go along, what I want you to do is make it go in a little bit. You see how I'm doing this here? I'm making it look like it's taking something in, swallowing it, and that's where I'm going to put my little dot. Could be a bacteria, could be a virus. In our case, we're talking about viruses. And it's going to ooze around that and swallow it. And then once it gets in, it's going to be inside an endosome. So there it is, inside an endosome. Now you're probably going, wait a second, that's exactly what the virus wanted. However, this cell probably doesn't have the right attachment. Remember, it has to be the right cell receptor. So if a macrophage doesn't express sialic acid or ACE2 or whatever it needs, the virus will be trapped inside there and it'll just get digested. And its parts will get recycled. So let's make another oozy arm. Eating something. And your macrophage can go off the paper too. Just make it bigger. It would have a nucleus, but we don't even have to put it in right now. You can just, it's there. So there's it eating something, and then there's some particles around. It's waiting for it. So it doesn't really know what it is. It's just going to eat it. When in doubt, eat everything you find. That's the rule macrophages live by. Don't ask questions, eat it. I'll draw a few more inside endosomes. Now the endosome can go merge with another kind of a bigger endosome. The macrophage has lots of big endosomes that are called lysosomes. I'm going to add a little bit of yellow to make sure we know those are special. Now, a lysosome is kind of like a stomach, digests everything. It's full of little scissor enzymes that can cut apart proteins, sugars, fats, nucleic acids. It's a super digesting machine. If you get in there, you're doomed. So what will happen is the little endosome of the virus will go and merge. They'll touch. It'll come over and it'll merge with a lysosome, and the virus will get dumped into a digesting lysosome. Actually, let's label this whole thing down here. Let's write macrophage. And then I'm going to point to this, right, lysosome. And then maybe in parentheses, digests. And if you already know what they are, you don't have to add notes. And of course it would have a nucleus with DNA, and it would have Golgi bodies. It has all the regular cell parts, but I really don't want to take time to draw them. Now, two more things to add here before we go on to our other cell. And first is find one of these endosomes to point to. I'm going to make another one over here because I have this space. These endosomes that are bringing things in, they happen to have embedded in the membrane, the wall around them, they happen to have the TLR, toll-like receptor 3. They have a viral sensor embedded in the membrane. So if they sense that that little thing they've swallowed is a virus, right away they're going to go and they're going to merge, take it to one of these lysosomes, merge the two together, which will result in the virus being dumped into the lysosome and digested. And we'll talk more about TLR3 in just a minute. Then the other thing I want to add is I want to add an MHC flag, but it's going to be a little bit different. We've been drawing that MHC1 like this, kind of like 4 squares with a little clip here and 1 flagpole. The macrophage has one that has 2 poles sticking in. Makes it look like one of those double popsicles you can buy in the summer. So it's got 2 flagpoles. Oh, and just to remember that they're not really squares. They look like this. Remember, they're a mess of tangled proteins, but we draw them as little squares, which is much easier. So let's put a little MHC flag. You have to draw it really tiny and make sure you get a second pole, 2 poles in there. And this is called MHC2. And this will allow it to show viral particles to other immune cells. Now, the other cell for right here, it's a lot like this. It just has skinnier arms. So it might look more like this. However, at the ends of some of these arms, wait, let me show you this one. The arms at the end there are grabbing, in this case, bacteria. See, it's grabbing a bacteria and it's going to suck it back in. And there's the bacteria. It ends up in an endosome. It's reaching out and grabbing the pathogen and bringing them in. It can also swallow them right like this, close to the edge. But sometimes it puts out these long branches. We call them dendrites. So you might want to draw a few arms grabbing viruses. Again, these will grab just about anything. So in a way, they're sampling the environment. They're just eating all kinds of things. This is a dendritic cell. And of course it's got a nucleus and all the parts. And let's give it some little viruses to eat. Now, how big would a virus actually be on this scale? Well, these guys, let's say approximately 50 microns. Do you remember in the lesson where we drew a big bacteria? I think it was lesson 2, where we had a bacteria and then how big it is compared to all the little viruses. So a bacteria is about 1 micron. So this guy's 50. So a bacteria would be like a tiny circle like that. And you remember how small the viruses were compared to that bacteria. So a virus would be a very tiny pinpoint, but we're going to draw them a little bit larger, dots that we can actually see. Okay, and again, they're going to bring it inside. And they also have that MHC2. We'll draw it really big here. They also have those. They would have a lot of them. 1 is enough. That's okay. And so what happens is these little particles that it brings in will get digested, and then brought back out on display on these little clips. So you could put a piece of viral protein. Now it won't be the whole virus. It'll be like a piece of the capsid, you know, just a little bit of protein that's been broken down, a little protein sample. So I'm just going to put a little line there and say viral protein. A piece of a broken capsid, or a matrix protein, or a glycoprotein. Very often they're glycoproteins. So these guys are about, you don't have to write this, but if you want to remember, 50 microns in size. So what's going on here isn't specific to any particular virus. These things just gobble everything in their path, whatever it is. So again, this action would be part of your non-specific or innate responses. So let's move over here and see another aspect of your innate or non-specific system. And that is these sensors. And we've talked about these a bit before. And you probably remember seeing TLR3 before. I think we mentioned that it's this area right here that recognizes the viral double-stranded RNA. And this sensor sticks to the outside of the macrophages and the dendritic cells. And it's also, as we just mentioned, found in the membrane that makes those little endosomes. So in the endosome, if viral double-stranded DNA is sticking here, it knows to get rid of that particle and go merge with the lysosome. Make sure your pencil is nice and sharp for this. We're going to do some pretty small little drawings in here, but nothing too difficult. So draw a little curve right about here. That's going to be the cell membrane, and TLR3 is just going to be this little curlicue thing. Right, so it's found on the outside. And then along comes a piece of double-stranded RNA. Now, even if viruses have a single-stranded RNA genome, which most do, still at some point in their replication cycle, when they're making copies of their genome, there will be some of the DNA that's stuck together. So there will be some double-stranded RNA, kind of as just a byproduct of replication. Now some of you might be wondering, what is viral RNA doing outside a cell? Why does it have these detectors on the outside? When the virus replicates, that happens all inside. So maybe this guy's next door neighbor's cell just died with a virus, and the stuff got out and it's oozing around the outside of this, let's say, macrophage cell. So maybe this guy's next door neighbor's cell just died with a virus, and the stuff got out and it's oozing around the outside of this, let's say, macrophage cell. So let's say this guy's been activated, and I'm just going to make a couple little curly things to show that it goes through a membrane. In fact, I might even make it 2 lines, so it's like a double membrane. It's got little anchors through the membrane, and then underneath, part of this molecule is going to be touching or be near a few other little proteins, and I don't even know exactly what shape they might be. We're just going to draw them as circles, and colors don't matter anything. We've got a yellow one, or I mean an orange one, and a blue one, or whatever. So there's some little proteins that when this thing touches, okay, it's going to cause a change in these proteins, and then that will result in a few other proteins changing their shape or doing something. We're just not even going to say what it is. I have no idea. Something. And then because that happened, these things are going to change their shape or be able to do something else. They're going to go affect a whole different set here. So you get like a chain reaction going. And they're going to affect these down here. And finally, the end result will be that a protein will go into the nucleus. All right, let's draw a really light line like that, and then make it look like this, where there's a little hole here, and a hole there, and a hole there. So this is the nucleus down here, and these are the pores. So something's going to go in to the nucleus, and then all the DNA is down here. And then some of the DNA will be red. One section of instructions will be red. And then out will come a messenger RNA. And that will be read by a hamburger bun ribosome. Right, so this is mRNA. And what will be manufactured is, I'm going to put IFN, interferon. The ribosome will make the interferon protein. And then the interferon will cause all kinds of other chain reactions to happen, all kinds of other proteins, antivirals to be made. So now we're going to talk about two other sensors, RIG1 and MDA5. And here's how their protein structures look. That is RIG1. And actually, usually they show it in diagrams. They draw it this way, and they draw this as kind of a little rectangle. And then they draw this as a line, a circle, and then you'll see in a minute how they draw this. They usually show it like that. And that is MDA5. And we already looked at a chart of what they do. Do you remember this? There's RIG1, MDA5. Now actually, see, MDA5 doesn't really look like that. These both are pictures of RIG1. But remember, they start this pathway, and that's a mitochondria. They interact somehow with this membranes attached to the mitochondria, and then there are these chain reactions. It does this to this group of proteins, and then this group of proteins acts upon this group, and this group. Oftentimes it's a little scissor of something that cuts one of these in half, and the little piece comes off, and that piece just happens to fit down here in these. And then another scissor comes along, and we have this chain reaction into the nucleus. This is what we just drew. And then it'll make messenger RNA. We don't see that here. We just see things coming in. And then there's this other pathway. It can take all these, and it can come in. The end result is the DNA instructions for interferon are read. So this is basically how we're going to draw RIG1. Not that complicated protein tangle. It's just a rectangle with two lines coming out, two smaller rectangles and a circle. We can handle that. And the way it works is, this is the sensor part. You see here's the double-stranded RNA. Sometimes there's 3 phosphates hanging off the end. That's a viral tag also. And so this thing is activated when this sticks. And then this coil uncoils, and the shape changes. You see it looked like that, and now it's going to be out straight like that. Although in our picture, we're going to draw it like this. It's going to fit in better if we draw it like that. But it doesn't matter, because this thing floats around in the cell. It's floating around the cytoplasm, and it can be turned any which way. So let's draw, we're going to draw it twice, just like it was twice in that diagram we just looked at. In its inactive form, it's going to look like rectangle, like that, and then curve over like that. And if you want to make them a color, you can. Now each little section here, each shape, would be what they call a domain. Remember, they hate the word area. They say this is domain. So they often make the domains different colors. And you can do any colors you want. It's not important in this drawing what colors you choose. So that's how it starts out. Make sure we know this is rig 1. Right, so which one we're drawing. And then along comes some double-stranded RNA. We'll draw it hitting the sensor down here. I'm going to draw the same thing again. This time we're going to draw the double-stranded RNA across the sensor. Again, double-stranded RNA, that's not specific to any virus, so this is non-specific defense. So I'm going to make my little protein showing through. And then the shape changes, it uncurls. So this causes the shape change, and then this shape change causes a whole bunch of other changes that lead to the production of interferon. Starts a signaling cascade to make interferon. You can just put IFN. So we're just not going to show MDA here. It would look a little bit different, but it would have the same effect. And this chart, just happen to notice, tells us that influenza virus's NS1 protein, that's one of those little tools it keeps in its capsid. That NS1 is designed to be a wrench to throw in this, to keep this thing from straightening out. So influenza can stop this little piece from springing out. And if the piece doesn't spring out, then this cascade never gets started, and then interferon is never made. So for number 4, I didn't know what to draw. I just listed interferon. It's one of these layers of defense. The sensors trigger the production of interferon, a chemical message that causes the production of many antiviral proteins, and also alerts other cells to the presence of the virus. And we've talked about that before. Also talked about the fact that like hundreds of little proteins are made and we don't even know what they all do yet. So I can't really tell you a lot about them because it's really hard to find information. And then number 5 is our last non-specific innate defense, and that is natural killer cells. So here we shift to an entirely different type of cell. So I'll show you a couple pictures how they look under the microscope. You could just take your average kind of microscope you use in a high school lab, or maybe you have one at home. You put some blood on the slide and you apply some stain, and you would be able to see these. Now remember, without stain, these aren't going to show up so nicely. But the ones that are stained purple here are the lymphocytes. These are the red blood cells, the ones that carry oxygen. I don't know what the little purple dots are, it didn't say. But these things are a type of white blood cell called a lymphocyte. And there's basically 3 types: T cells, B cells, and NK cells. And you can't tell the difference. So this lymphocyte could be any of them. So to kind of simulate the way they look under the microscope, we can kind of draw something like this. And I believe this is basically the nucleus. The nucleus takes up most of the cell, and it would usually be darker, but you know what? This is going to be a cartoon picture, so we're just going to leave it like that. And this is going to be a silly cartoon, so we're going to give it eyes looking down like that, and then eyebrows that are kind of a little bit disgruntled looking, and a little bit of a He's very serious. He's not really angry, but he's concerned. And then NK cell. I'll put freckles on NK cells because they're a strange kind of lymphocyte. They're a granular lymphocyte. So these little granules here are going to turn out to be in the picture in a minute. And I'm going to write NK for natural killer. So what is the NK cell looking at? Looking at the surface of a body cell. So right below here we'll leave a, well actually maybe we should just go ahead and draw a little hand here. Okay, so the cells don't really have hands. This is cartoon. Draw just sort of like this, a little hand as if it's feeling a surface. And the surface it's feeling is the surface of a cell. So kind of make a little line going like that. And then it's going to have another little hand right here. You can see I drew it and then erased it. I think it's better to draw what it's feeling first. Let's draw the flag first. I drew the hand first and it didn't work out so well. So right here we're going to put one of those MHC1 flags. That. And I'm going to put a little bit of, it's holding a little bit of viral protein. So the virus went inside and its little pieces got chopped up. Remember there was a virus that tried to keep these from going to the surface? It was the herpes that kidnapped these. Because when they go to the surface, guess what? This guy, draw a little hand type thing going like this. It can feel, it feels the MHC1. It knows that it's attached to a body cell and it can feel this little bad viral protein. Now, herpes thinks it's going to trick the NK cell and just kidnap all of these. And if the flags never go to the surface with viral proteins in them, then the NK cell will never know. Right? It'll never see the bad viral protein. However, the NK cell can also feel the absence of flags. Can feel absence. So you can't fool him. The virus says, okay, fine, we're just not going to put those flags out, so you will never know. And it goes along and says, no flags? I think I know what's going on. Yep, something bad's going on inside, and it will kill the cell. So if it feels a bad protein, it's going to kill the cell. And if it feels no flags, it's going to kill the cell. Now, some viruses try to get around this by having the cell put up fake flags. And that's really complicated and a whole other story, but I think it's interesting to know that the virus will even try to get around this. So first, why is it going to kill the cell? Well, once a virus gets inside and starts its nasty work, there's really nothing you can do. There's no way to just clean out the cell. The cell's doomed. So the only solution in this case is just to kill the cell as quickly as you can, as soon as the virus gets in there, before it has a chance to replicate. And then healthy cells in the environment, they'll go through mitosis, they'll duplicate, they'll make copies of themselves, and they'll just fill in that empty space, and the cell will be replaced. Now, how does it kill? Well, it has a really, really nifty weapon. You'll think I'm making this up. This is so cool. We're going to give it sort of a third hand here, or I don't know, a little holder or something. Draw a little thing coming out like that, as if it's a hand or a finger or something. And then we're going to draw basically a little gun. It's called perforin, but I call it the perforin gun. So just draw kind of like an L shape like that, and then like that. So the NK cell shoots little bullets at the cell. But what they do when they come out is they will make a hole. So make your little bullets, and it doesn't matter what color you use. It doesn't matter at all. As they hit the cell, and now we kind of have to make them going down because of the way our drawing is, but it would probably just aim it right at the cell. But We just have an awkward space to work with. So as they come out, the proteins are going to assemble themselves automatically. They'll just lock together to form a ring, and they'll actually make a hole. And it'll look something like this. Here's a cross section. So there's a hole right down through there. And there is an actual picture. There's some pictures of some rings looking straight down on it. And there's a little map of what the protein looks like. And there's a picture up here of how they fall, go into the membrane, they start locking together, and more of them lock together to automatically form a circle. And of course then that's what it looks like when it's done. So that's the first part of the operation. So make them form kind of a ring like this. Sort of a flat oval. And then along with these perforin proteins, there's something else too that comes out. I'm going to use red so it looks bad and it shows up well. And these then go through the hole and these are the thing that actually kills the cell. Well, the hole will a little bit because it might leak some stuff out. But these things, I'm going to put a red dot right here for my key. This red dot equals something called granzymes. G-R-A-N, gran, and then zyme, like the word enzyme, Z-Y-M-E-S. Granzymes cause cell death. Now how do they do this? Well, these will go down into the nucleus and it's going to start a chain of events which will end with the DNA being shredded. You know, we have little proteasomes that shred proteins. Well, there's a little machine that will shred DNA. Now this is a natural cell process. The cell has this equipment waiting if it ever needs to use it. And so the granzymes say, start the process right now, you're done for. And the shredder will go to work, and then without any DNA, the cell can't do anything, can't make proteins, and then the cell gradually just shrinks and shrivels up and disintegrates. And then a macrophage comes along and gobble gobble and eats up all the debris left over. And the macrophage will recycle the parts and make them available for new cells to use. So let's label this gun perforin. I call it the perforin gun. They don't use the word gun in science books, but I just can't help but think of this as the perforin gun. So now we meet some of the members of the adaptive or specific immune system. And it's specific because these cells can recognize individual pathogens. They can tell the difference between coronavirus and influenza virus. And the ones we're going to meet here in number 6 are T cells. Now it says here killer Ts, but we'll find out the killer T is going to need help from a helper T. But we can't get our story started until we have a body cell over here. So let's make the edge of a cell. And let's give it some MHC1 flags. Make sure we have one sticking right out here where that T cell is going to be able to feel it. MHC1. Body cells have 1, macrophages have MHC2. And surprise, surprise, the clips are holding some little bits of bad viral protein. So we'll draw this T cell that'll be able to detect that. Now all the lymphocytes have a huge nucleus. So our cartoon is going to have a big circle face like that. And since it's a cartoon, we're going to give it eyes looking over at its buddy there. And then we make eyebrows kind of going up like that. And a mouth that's just kind of a slanty line like that. So it kind of looks confused, like it doesn't know what to do. It can put a question mark there. And what he's confused about is, should I kill? And let's write T with a little c. Now, they like to use the word cytotoxic now. Everyone used to say killer T's, and a lot of people still do say killer T's. I mean, I wrote killer T's. But the correct name is cytotoxic T cells. So since the word cytotoxic begins with the letter C, that's how we show which type of T cell this is. It's a T with a little c for cytotoxic. Now T cells have a type of receptor that no other cell has, so we call them T cell receptors. And I'm going to make mine just look like a little Y thing. The receptor has a particular shape on the tip, and every T cell has a different shape. So you've got millions of T cells in your body, and each one has a receptor that has a slightly different shape. Now most of these shapes won't match anything. Most T cells will never be used. Once in a while, there's a T cell whose receptor shape just happens to match exactly a little bit of pathogen protein, in this case a little bit of viral protein. Just coincidentally, these happen to match. Well, it turns out that this is a killer cell, so it's got, I'll give it a little, I don't know, give it an extra arm or hand, a little, if you want to give a little finger. It happens to have a perforin gun. So here's the gun kind of in its holster. Now, of course, T cells aren't complete organisms. They don't have a brain. It doesn't know anything. So there's a system in place for it to check to make sure that that's a bad thing and that should kill the cell. So it sort of like needs to get permission from a helper T cell. So let's make this guy look just the same, but he's going to be T with a little H for helper. And he's looking over at the killer cell. And maybe he's got a little bit, he's not really evil, but it's like, you know, he's like, yeah, we need to kill that thing. Put an exclamation point. So let me show you a drawing that's kind of indicate what we're going to do in this space. So these immune cells can talk to each other various ways. They can release cytokine messengers, but there's a whole bunch of little places where they can touch. They have receptors, and so I thought we'd just do kind of a non-specific little drawing here to show that they're communicating. Okay, so they're communicating with each other. Now this happens to be a T helper cell that recognizes this same shape also. So it's recognized this shape, and the reason that it knows that this shape is around is because it sensed it on the outside of a macrophage. So it came along in another part of the story, and it sensed a macrophage holding this little bad purple protein. And he says, oh yeah, I saw that bad thing you're holding. I saw that on a macrophage. And I know there's an intruder lurking, and I think you found it. So yeah, the macrophage told me to be aware of that and to be on the lookout. And if I found someone with this match, go ahead and tell them to kill. And so because it was given instructions by the macrophage to tell this guy to kill, it can send a message. Let's make it a chemical message, some cytokines. Chemical message goes across. Okay, it's a message that gives permission to the T cell to get out its perforin gun, blast a hole in the cell, and put the granzyme. So this guy does exactly this. So you don't need to draw that again. We know what the perforin gun does, all that. So we know that's what will happen when he shoots the cell. So now we meet the last type of lymphocyte, the B cell. So there's three, there's NK, there's T, and there's B. They all look very similar, so this is going to actually be a T cell, a helper T, and this is going to be our B cell. And we're going to have a macrophage over here. It's kind of coming in over here, and one of his blobby arms is going to be sticking out like that. So this will be our macro. Now, since we're drawing cartoons, if you want to put little eyes on it, make a blobby ghost, you can do that. And it's presenting or showing its MHC2 clip to this T helper cell. And guess what it's found? It gobbled up some viruses, digested them, and it's going to show this helper. So let's give him a big round face. Give him some cartoon eyes. He's actually going to be looking over here at this other cell. Alright, he's kind of going to be in a very serious mood here, serious business. And he is a T helper. Now there's different kinds of T helpers. So the T helper we met up here specializes in this job. And this T helper down here is going to specialize in talking to B cells. So let's label this MHC2. Right, so it shows this to the T cell who has a little receptor to feel it. Exclamation point. Okay. Instructions. And then there will be some cytokine instructions to go over to help it know what to do. Then our B cell is going to look very similar. It's hard to tell the difference under the microscope. They look the same. Looking over at the T helper, kind of looking like question mark. So what is the B cell asking? Oops, let's put a B there. Well, the B cell isn't asking, oh, should I go kill with my perforin gun, because it doesn't have one. What the B cell has is something called antibodies. We're going to draw them looking like little Ys, and the reason is because they look like Ys. And you can use any color you want. Make some little Ys, these little antibodies, sticking off. And this B cell can only make one shape. Every little type of antibody inside this Y, the top part, the forked part, it's like this, where it'll only match one thing. It's a very particular shape. And so some B cells will just happen to have the right shape to fit a pathogen, in this case maybe a virus. So most B cells, their shape won't match anything, and they'll sit there in your body their whole life and never get used. Out of all the millions of different random shapes, only a few will end up matching the pathogen. So he's like, well, this is what I can make. I can make these Y-shaped things. They say, do you want me to make a lot of these? And so this guy will say, well, you know what? If your little Y matches this thing, then yes, I do want you to make a bunch of them. So if that happens, let's make an arrow. If the B cell gets permission to make a lot of its little antibodies, it's going to start cranking out these little Ys. Now, why is this a good thing? Well, we did mention that this shape might match a viral protein. So right here, what we're going to draw in our last little corner, we're going to draw a virus, sketch in a circle right about like this. This is going to be our virus. And we're going to need to leave room for a big eater macrophage to be coming in like that. So I think I need to leave a little more room. Maybe I'll make my virus a little more. So I always draw lightly so I can change my mind. Put the virus out more like this. Okay, so I'm going to put a big V on it for virus. And then I'm going to give it little glycoproteins. Very quickly, I'm not going to take any time to make them look very good, just like that. And these little Ys are going to go and stick like this. The inside of the Y, the fork part, is the part that sticks to things. You see the bottom part sticks to the cell, but then when they stick to something else, it's the top part of the Y that sticks. And so they're going to go and stick to the virus. Now this will accomplish several things. Number one, is the virus going to be able to stick to a cell now? Right now, these things are going to block it. It's going to clog up those glycoproteins and won't be able to attach and enter a cell. And second, these little antibodies are like yummy little candies to several body cells, including macrophage, which we'll draw here. Okay, so we're making cartoons, so if you want to, we can put an eyeball on. It looks like a mouth, right? Okay. And it's like this big, kind of shaded a little bit, so it looks like a blobby monster coming in here like this. And it's thinking, yum, yum. And will eat anything with those tags. So that is hopefully the end of the story for all viruses in your body. If they get through all those barriers, they don't get trapped by any of the innate system, then hopefully this will kick in. Now this takes some time for this to happen. This takes days for this system to kick in. But eventually, hopefully, your T cells, B cells, and macrophages will catch up with the virus, and all of them will be eaten by the macrophage, who will then put them into the lysosomes, dissolve them, and chop up the little parts. All the proteins will get chopped up, the viral RNA will get chopped up, and then the little pieces will be able to be recycled. And so some of your cells are made from proteins and the RNA that used to be part of a virus.